Good morning and welcome to the first session on innovations in clinical trials. I am Rob McMaster, now serving as the past chair, Clinical Trials BC Advisory Council and Vice Dean of Research, Faculty of Medicine at UBC. We have 6 outstanding presentations in this first session that will discuss advances in clinical trials, including Alzheimer's disease, multiple sclerosis, pediatrics, chronic kidney disease, telehealth, and lung health. Our second section this morning will focus on innovations in cancer clinical trials. I am delighted that our first speaker will be Dr. Howard Feldman, currently Dean of Alzheimer's and Neurodegenerative Research at University of California, San Diego. Howard is very well known to us in BC as he previously held several positions. He is internationally recognized as a world leader in Alzheimer's disease research. And today's presentation will be on adaptive clinical trial design in neurology. Our second speaker will be Dr. Tony Trablusi, Director of the Multiple Sclerosis Research Program at UBC Hospital and a world leader in MS clinical trials, where he was one of the first to apply MRI imaging to study the complexities of multiple sclerosis. His presentation will be entitled Inclusion of Imaging in Clinical Trials. Many clinical trials involve hundreds, if not thousands, of participants. However, rare diseases present an entirely different aspect to clinical trial design, where there are only a few patients worldwide. I am delighted to introduce Dr. Sylvia Stockler, Head of the Division of Biochemical Genetics at BC Children's Hospital. She will be presenting on the innovative approach, knowledge base, N of 1 clinical trials. As our fourth speaker, we're very pleased to have Dr. Adira Levin. She established and is the scientific director of the CIHR Strategies for Patient-Oriented Research Network, CAN-SOLVE-CKD, with extensive patient partner involvement. Today, she will be presenting on networks as an innovative approach to clinical trials. The fifth speaker will be Dr. Kendall Ho, who is widely recognized for his innovations in telehealth for both research and clinical care. Kendall is a practicing emergency physician at Vancouver General Hospital and the academic director of the newly launched UBC Cloud Innovation Center. Today, Kendall will be discussing the most timely topic, telehealth innovations in clinical trials. The last speaker in this morning's session will be Dr. Dawn Sin, Director of the Center for Heart-Lung Innovation at Providence Healthcare and one of the world leaders in COPD research. Dawn will be presenting on innovations in lung clinical trials. We look forward to an exciting first session on innovations in clinical trials. Thank you.
Good morning. I'm Howard Feldman at the University of California, San Diego, where I direct the Alzheimer's Disease Cooperative Study, a large dedicated national initiative to the conduct of clinical trials in Alzheimer's disease. Here are my disclosures. And here are the 3 objectives for this morning's short talk on adaptive clinical trials. I'm going to start by providing an overview of how adaptive designs can be applied within clinical trials. I'm going to support this with some real-life example of neurological trials that I've been personally involved with that have deployed adaptive design successfully in the areas of migraine and Alzheimer's disease. I'm going to conclude by presenting the limitations, pitfalls, as well as advantages of adaptive trials. In these figures, we can differentiate a traditional fixed sample design from an adaptive design. Traditional fixed sample designs are familiar probably to everyone who's involved in trials attending this meeting. An adaptive design, however, is different. It allows for prospectively planned modifications to one or more aspects of the trial based on incoming accumulating data from trial participants. These reviews and adaptations are pre-specified. So as you can see in this lower panel, there's this additional layer where incoming data is reviewed, set up for modeling and Bayesian simulations, which lead to adaptations according to pre-specified rules, which then inform the further adjustment in the conduct of the trial on its way to the primary analysis. This stage of intermediate data analysis, modeling, and simulation is not part of a traditional fixed design. There are a number of ways that this may be manifest within design choices, and I present these pictorially in this slide. In the first instance, adaptive clinical trials with sample size reassessment refers to a circumstance whereby an interim analysis identifies that the sample size may be insufficient. The grounds for this may relate to increased variability of the primary outcome measure, more so than had been initially planned for, or alternatively, effect sizes look smaller than those that had been predicted, thereby requiring an increased sample size to maintain the power for the study. Notably, there is no change in the groups or study population characteristics. The second adaptation is adaptive randomization, guided by response. We can think about this as a circumstance of playing the winners or dropping the losers. As you see in the cartoon, there's a burn-in phase for the first part of this trial, and then simulations begin with incoming data. In the figure, you can see that the allocation favors enrollment to treatment 1, and randomization in turn is adding more persons by algorithm to treatment 1 and fewer to the losers, which would be the other arms of the trial. There's additionally an adaptation that can be made through an enrichment design whereby randomization balances prognostic factors. As you can see in this design or in this cartoon, there's an increase in the number of pink participants that represent a prognostic factor that looks promising within the first phase of the trial, and therefore the trial adjusts its randomization to balance and enable this to be looked at a later point in the trial. Not shown in this model-based slide is a continual assessment adaptation. This is based exclusively on a priori modeling and then incoming data, whether it's on safety, dose, biomarkers, or pharmacodynamic measures that allow contrast with what the modeling predicted and adjustment accordingly. And I'll provide an example of this one as well. Let's begin with response adaptive dose finding, and I use the example of rimegepant in acute migraine, a development program I was involved with while employed at Bristol Myers Squibb. Rimegepant is a calcium gene-related peptide antagonist, a so-called CGRP antagonist. This was the first trial that was undertaken, and we chose to do a phase 1-2 mixed design, double-blind, multi-center, randomized trial evaluating efficacy and safety including placebo, active control with sumatriptan, and 6 arms of rimegepant in acute headache using 2-hour pain-free as the primary outcome measure. The research plan called for adaptive dose finding across the 6 single-dose levels of the drug. Playing the winners was achieved through dynamic allocation based on incoming data of the primary endpoint. Early stopping rules for either success or failure were also in place and running through the Bayesian simulations. The way the trial was actually conducted was that there was a burn-in phase with initially a fixed group parallel design, 336 participants, with a fixed equal allocation to each active arm. Beyond this first 336 participants and through to its final sample size of 885, randomization changed from being fixed to blocks of 8 participants, 1 to placebo, 1 to sumatriptan, and then per the Bayesian algorithm, dynamic allocation to active arms. Here's what the study looked like through its conduct to its completion. I draw your attention to 10 milligrams and 25 milligrams, which have notably fewer participants complete, as it was identified through the algorithm that these were less likely to be successful. By contrast, 75, 150, and 300 milligrams were felt by the algorithm likely to be the winners and stood out for having significantly better outcome than the placebo group. Fast forwarding, 75 milligrams has made its way through successful phase 3 development and is now becoming a marketed drug. Here's an example of a sequential dose model-based continuous assessment. This is a seamless phase 2A/B design that we're currently undertaking at the Alzheimer's Disease Cooperative Study. I draw your attention to the adaptive dose finding in phase 2A. As you see, there are 3 cohorts that we've planned to test different doses. What is of interest here is that if cohort A can successfully be titrated, and if it does not meet stopping rules, it will inform a final decision of dose to phase 2B, and there will be no need for cohort B and C. The highest successful dose will have been achieved. If, however, it meets stopping rules for reasons of adverse events pre-specified, there's a provision for a drop-down adaptation to cohort B. If cohort B is not tolerated and meets stopping rules, there's a drop-down to cohort C, which is the lowest dose that will be tested. If cohort C is not tolerated, the study will be stopped. The successful dose of phase 2A informs phase 2B, and there's a variety of stage gates and rules about what success and what the green go criteria are. Thinking about the value and adaptive and pros of adaptive trials. Planned flexibility. So rather than fixed design, there's an opportunity to be flexible. Program efficiency, optimal sample sizes, less opportunity cost, more exposure of participants to effective doses, and decreased exposure to unsafe or ineffective doses, and quicker decisions for safety, futility, or efficacy. The examples I provided show some of each. There are some cons. Regulators are somewhat skeptical and need to be convinced of the value and the tightness of the modeling and simulations that are being used to support the study. The designs are complicated. They require active management throughout their course. The logistics are more difficult. There's a payback on this, but there is a cost. Statisticians have concerns, as do others, about type 1 errors and how this is controlled, how confidence intervals are covered, and how treatment effects are estimated. These are all points that have to be very carefully considered within such designs. And as always, adaptation may introduce unexpected bias or variation. I acknowledge my colleagues at the Alzheimer's Disease Cooperative Study, project directors, and those that have been part of this initiative that has helped test some of these adaptive trials. Thank you.
Hello, it's an honor to be included in this conference. I'm very excited about the leading role BC researchers can undertake in clinical trial innovation. And as a case study, I'll focus on my experience as a clinician scientist and imaging expert in multiple sclerosis. I've had many local and global roles in research involving phase 2 and phase 3 clinical trials over the past 20 years. When I joined the UBC MS Clinic in 2001, I began working with an imaging research group to develop MRI protocols for multi-center international clinical trials, and we collected and analyzed hundreds of thousands of MRIs from around the world. That analysis was key for the approval of the very first treatment for multiple sclerosis and for the dozen or so treatments that subsequently became available. Through these multiple roles, I've seen opportunities and challenges in bringing new innovation in imaging outcomes to clinical trial use. I'm going to focus on some successes and challenges as an imaging center and also as a principal investigator conducting a local imaging sub-study. A little background first. MRI imaging of multiple sclerosis became clinically available in the 1980s at UBC, and we had one of the first scanners in Canada. MRI had revolutionized neurology and neuroscience research, and locally, Professors Don Patey and David Lee realized early on the potential MRI had for monitoring patients, particularly for detecting clinically silent disease activity. What you can see on this image that's cycling through on the right side is one patient, and the white spots showing up in the center of the screen are new lesions of multiple sclerosis. And these occur in the absence of clinical symptoms and can occur at a ratio of about 10 new MRI lesions for every clinical attack a patient has. This therefore becomes a very powerful clinical trial outcome. It represents the inflammatory aspect of multiple sclerosis, and we can design studies to detect a therapeutic impact on preventing those new lesions, which is much smaller sample size than if we were relying on clinical relapses alone. And here's a typical clinical trial outcome. Four different treatments shown here. One's placebo, another is comparative medication, and then 2 doses of a new drug. And within 8 weeks, you can see a significant impact on preventing new MRI lesions from showing up. That's sustained up to the 3-month marker, week 24, rather the 6-month mark. This really decreases the amount of time patients need to be exposed to medication to determine if a drug should go forward to a phase 3 clinical trial. While we're still using the same new lesion metric in clinical trials, further innovation in design allows us to even decrease the number of patients and shorten the duration on study or the placebo exposure, which is really important in this current era where there are so many treatment options for patients. It's hard to enroll patients into a placebo-controlled trial that's lasting more than a few months. And this current design uses statistical modeling called multiple comparison procedure with modeling. And by only using 4 weeks of placebo exposure and a 4-month study duration with multiple doses of a new drug, we can model the lesion response to the treatment and find the best dose depending on how the therapeutic response is on these different curves. And this has been a very popular design with patients. In the most recent study we did, only 1 patient had dropped out of this multi-center trial, which is quite remarkable in the phase 2 study. Our imaging group that started in the 80s quickly became the world leader for central reading or central analysis for clinical trials. And that success brought many challenges that can conflict with academic aspirations. Specifically, to run a analysis group for clinical trials, we need a large investment in infrastructure to meet those regulatory and business demands. That could often lead to more of a rigid analysis pipeline. You'd have to lock down your procedures for approximately 5 years for the duration of the study. And that would decrease the opportunity to modify those techniques to improve their outcomes or their ability to detect new ideas around the pathophysiology of MS. And while we did accumulate hundreds of thousands of images, which would be ideal for looking at machine learning or artificial intelligence research, that data is only useful if you have the clinical and treatment information, which is often kept separate by the sponsor and can create quite a long bureaucratic negotiation to get access to that data. And so without, in parallel, investing in innovation, it can become quite rigid and difficult to develop new techniques while maintaining a large central analysis group. So what I want to switch to now is an area where we've been able to use local imaging sub-studies as part of a multi-center clinical trial to advance local research interests and development. And this has been a very exciting and promising area that in ways is less stressful than trying to run a large central analysis group for multi-center trials. So in this video, which is going to cycle through a few times, you're not seeing many new lesions show up in this MS patient, but as you can see, the brain is changing its shape over time. It's shrinking. The ventricles, the white parts in the center, are getting larger. And this concept of brain atrophy or shrinkage represents another aspect of pathophysiology of MS where there's a degenerative component that is somewhat independent of new lesion development. And that's one of the biggest challenges we have now in developing new therapies for MS and in neurology, is trying to find biomarkers with imaging that could best reflect the pathophysiology of what's driving this degeneration. Locally, there's work by one of our physics researchers in the Division of Neurology, Shannon Cullen. She just received the UBC Department of Medicine Excellence Award in Basic Science Research for this work in myelin imaging. And here's just an example of how we can use this technology. You can see in gray are two different patients with a standard clinical MRI that looks quite similar, with a few lesions around the ventricles or the center part of the brain. With this technique called myelin imaging, using the MRI, she can look beyond the lesions, which here show up in red, and look at the general health of the myelin throughout the whole brain. And the darker regions, particularly the patient on the right, are areas that are grossly demyelinated. And that really reflects with the greater disability that patient on the right has. And so now we can add to a piece of knowledge, lesions, more data on the health of the rest of the brain to better monitor disease progression or repair. And that's what we've adapted into a clinical trial. So here's an example where in a very small cohort, we were able to do a sub-study at our center. We had close to 60 patients participate in the study. It was a phase 3 clinical trial. Half the patients were randomized to an older drug called interferon in red, and the other half were randomized to the newer therapy called ocrelizumab in green. And we had a control group. And over 2 years, what we could see with this biomarker is that the control group, the measurement's stable, which gives us confidence in the measure. But the patients on the older treatment did not do as well. Their values were starting to decrease or demyelinate, whereas the patients on the newer therapy in green were stable or improved. And so this hypothesis generating approach with a sub-study allows us to work with a relatively small sample size to demonstrate benefit of a new biomarker. We can even translate that into individual patient metrics. Here's a lovely example, again, with one patient on the left with the older treatment and the other patient on the right with the newer treatment. Areas that are yellow or red are very severely injured or demyelinated regions. Areas in green are regions that were demyelinated before starting the treatment and have actually improved over time. So not only can we use this technology on group level, but we can also potentially use it for individual patient care. So for this type of innovation to be successful, you need a successful clinical trial unit and patients that are very engaged in the participation of studies, and then the collaboration between scientists and clinicians to work together to come up with these sub-study ideas and to promote them towards the sponsor. And we've had a lot of success with different companies doing this type of approach. There's a major impact of this type of innovative local sub-studies. For example, just giving an overview of multiple sclerosis impact, over the past 10 years, we've had about $20 million worth of clinical trial contacts. And on top of that, for local imaging research funding, about another $9 million, which is quite impressive, really supporting the development of and training of new personnel. So I'd be happy to talk about this forever, and I look forward to the discussion. Thank you.
My name is Sylvia Stockler. I'm a biochemical geneticist at the Department of Pediatrics. And I would like to talk about evidence generation for treatments of rare diseases. And I will be using inborn errors of metabolism as an example. Inborn errors of metabolism are the first group of genetic conditions with causal therapies. We already have 3 generations of therapies. The first generation, which goes back to the 1960s, includes medical diets for disorders of amino acids, carbohydrates, fatty acids. The second generation includes enzyme replacement therapies and small molecule therapies for lysosomal storage disorders. And the third generation includes gene therapy and precision medicine therapies for individual patients. When we look at evidence, there are some challenges, though. Among the first generation therapies, in more than 60%, only non-experimental evidence is available. Still, most of them are considered standard of care. Among second generation therapies, we do have experimental evidence. However, there is a discrepancy between experimental and real-world evidence. And this is mostly due to the fact that surrogate, rather than clinical and personal meaningful outcomes, have been chosen as endpoints in the clinical trials. Among the third generation therapies, we do have challenges regarding post-marketing evidence and employment of innovative trial designs. There are also lots of opportunities to create evidence. Guideline developments will lead to standardization of first generation therapies as a baseline for future clinical trials. Alternative trial designs can mainly be used in situations where you have small patient numbers and multiple treatments available at the same time. Registry-based studies can be used in non-experimental and in experimental settings. And N-of-1 trials definitely will be the tool for individualized precision medicine in the future. During the next couple of minutes, I will mainly talk about our work with registry-based studies and with N-of-1 trials. As an example of registry-based non-experimental studies, I would like to use pyridoxine-dependent epilepsy, which is a neonatal epileptic encephalopathy which has been known for quite a while. It causes seizures responsive to pyridoxine. However, 80% have developmental intellectual delay despite seizure freeness. The genetic defect was localized in lysine catabolism in 2006. And subsequently, our group was able to show that nutritional lysine restriction leads to a reduction of neurotoxic lysine-related metabolites. However, the crucial question remains, does nutritional lysine restriction also improve clinical outcomes? We first took an experimental approach and designed an open-label, negative controlled cohort study to investigate the efficacy of lysine restriction on neurodevelopmental outcomes in children treated within the first month of life. We came up with a sample size of 18 test and 22 control subjects in order to achieve an effect size of 60% reduction of neurodevelopmental disability. However, there was a feasibility problem. Given the very low incidence of this condition and a reasonable study duration, we would have had to recruit our study participants among 8 million newborns, which corresponds to a catchment area, including the United States, Canada, and the EU. We finally came up with a registry-based observational study. We recruited an international PDE consortium and created a longitudinal observational database, which is available online. This database allows us not only to study the natural history, but also the effect of lysine restriction and other treatments that have been developed since then. By 2020, over 100 subjects have been enrolled. As an example for a registry-based experimental approach, I would like to briefly introduce our Inform Rare project. This is a CIHR multi-year grant with Beth Potter from the University of Ottawa as the PI, and with Canadian Inborn Errors of Metabolism Research Network leaders and patients as co-investigators and collaborators. We will develop the basis for registry-based randomized trials for 3 rare conditions, namely spinal muscular atrophy, phenylketonuria, and mucopolysaccharidosis. As an example of N-of-1 trial, I would like to talk about our drug repurposing project for SLC6A8 or cerebral creatine transporter deficiency, which causes intellectual disability and epilepsy. We have started a virtual drug screening project in collaboration with patients, with our research institute, with UBC, and with Indiana University. We have selected 20 mutations present in patients known to us who can participate in the future in vivo candidate validation, which will be done via N-of-1 trials. N-of-1 trials lend themselves for customized proof of principle studies. They enable the comparison of on and off within one individual. Sequences of on, off, and wash out, so-called periods, can be repeated several times depending on the needs. Randomization and crossover designs are possible. N-of-1 trials have the beauty that we can employ many types of outcomes in these studies. Core outcomes, which are true for all patients and allow meta-analysis. In our case, this will be intellectual disability and brain creatine. Personalized outcomes are true for single patients depending on their phenotype, severity, and age. In our case, it will be seizures, movement disorder, and constipation. Personal meaningful outcomes are true for single patients under special circumstances. For example, adaptive functioning, behaviors, and sleep. In order to make N-of-1 trials amenable to a larger group of users, we have engaged in the development of an N-of-1 trial platform. We are currently garnering interest from collaborators, users, and beneficiaries to help us create the following deliverables: templates for trial designs and outcome measures, advice on regulatory, legal, and ethical issues, statistics, creation of a database, and finally of a platform with all tools available. All this work would not be possible without many other collaborators. What I said about evidence for inborn errors of metabolism recently has been published in the Journal of Inherited Metabolic Diseases. And here you can see all the collaborators involved in our drug repurposing project and in the N-of-1 platform project. With this, I would like to conclude my presentation and thank you for your attention.
Thank you for the invitation to present our network as an innovative approach to clinical trials. And my privilege is to represent a network as one of these innovations, specifically the CanSolve CKD network, which is Canadians seeking solutions and innovation to overcome chronic kidney disease. And it's on their behalf that I'm actually presenting this. You will recall the Strategy for Patient-Oriented Research, which is a coalition of the federal, provincial, and territorial partners that were dedicated to integrating research into care. and specifically to foster evidence-informed healthcare, bringing innovative diagnostic and therapeutic approaches to point of care. But really what we've done in CanSolve CKD is create this national network that brings together kidney patients, researchers, healthcare providers, policymakers, and caregivers to actually embody this vision. And our specific vision for kidney care is that every Canadian with or at high risk for chronic kidney disease will receive the best recommended care, experience optimal outcomes, and have the opportunity, importantly for this symposium, to participate in studies with novel therapies, regardless of their age, sex, gender, location, or ethnicity. So this pan-Canadian network has 18 research projects, over 60 patient partners, and 400 team members. And I'm going to tell you a little bit about that network as we go forward. Fundamental to this network are two patient-led councils. One, the Patient Governance Circle, and the other, the Indigenous Peoples Engagement and Research Council. Specifically, the latter highlighting the disproportional impact and prevalence of CKD in Indigenous populations. That foundation has with it a number of committees and research projects under a lean governance structure accountable to the funders. Our Patient Governance Circle embodies equity, diversity, and inclusion and was founded in 2016. These are people who are care providers, people living with different types of kidney disease at different stages of kidney disease, either caregivers, caretakers, family members, those with and without kidney transplants, on and off dialysis. Our Indigenous Peoples Engagement and Research Council has over 20 members now, including not only Indigenous patients and their families, but care providers, Indigenous researchers and scholars, as well as community leaders and physicians. And together, these people help to keep us grounded in executing our three research themes, which are earlier diagnosis, better treatments, and innovative care. And as I said at the beginning, there are 18 different research projects across those themes that use a variety of study designs and methods to answer questions. I've put on the right-hand side of this slide simply some key questions about identifying and treating early kidney disease, BET methods, how you delay progression with new treatments, a systematic review and review of looking at how we improve symptoms for those on dialysis from both the basic science all the way through to translational implementation science and improving living donor rates and experience. What we do in this national network with the structure is consolidate the expertise and resources in kidney research. And by so doing, we enhance the capacity, really focused on patient engagement and a development of some novel infrastructures, which I'll share with you now. So patient-informed activities are the core of SPORE, and what we've done is have support with patients for the development, vetting, and follow-up of clinical studies. We have formal priority setting processes. We have patients on our Research Operations Committee on every single research program, and also within the Canadian Nephrology Trials Network, which I'll also tell you about in a moment. We've supported the dissemination of information about trials through novel structures, one being Kidney Link and the other CNTN. Kidney Link is a new online platform that connects users to new research opportunities and curated educational experiences. It increases people's ability to participate in research. And through Record, another project or infrastructure, we help researchers recruit greater numbers of interested patients, both to be on their committees and research teams, but also to participate in trials. The Canadian Nephrology Trials Network is a pan-Canadian network that's striving to really improve multi-center research, specifically traditional clinical trials, but really to foster a culture of collaboration and then offer resources to help places around the country really participate and overcome issues with recruitment. We have opportunities for training. We support training and mentorship of patient partners through Kidney Pro, and also young investigators through Crescent, a national curriculum and training program for basic and clinical scientists throughout the spectrum of biomedical and non-biomedical researchers, really all asking important questions in kidney disease. Kidney Pro is an online learning module that explores the breadth and depth of kidney research in Canada, and it allows patients through learning to be able to figure out how best to participate in kidney research, and is thought to be a sustainable and renewable resource if we can get more and more patients understanding things. We have integrated very much cultural safety into our research activities, and our IPERC group has helped us with having a structure and have taken the lead in a curriculum development called Wabishki Bishnu Skanj Learning Pathway, which is actually an attempt to educate both researchers and patients in Indigenous learning ways so they can be better equipped to participate in research. In addition to that, we have tools that enhance patient engagement, how to tell stories with impact, and integrated knowledge translation. And what we've learned is that patients have really good questions, and that we can inform clinical trial design with patient input, and that might actually help us get people enrolled in the studies. We know that lay summaries are really important to facilitate engagement and understanding, and that most importantly, that people want to participate in research, but we've learned through them that many of our processes are quite prohibitive, and that's a problem. Not all patients like the idea of a randomized controlled trial. They think it's have and have-nots, and many have suggested to us that alternative designs might be more acceptable, especially in specific communities, such as stepped wedge design as an example. Our consent forms are far too long and don't make sense to most of us, and let alone to the people trying to sign them. But most importantly, our patients have told us they want to help answer important questions, and that has kept the network going. There are many ways to judge successes of networks, and certainly are conventional ways to look at things like publications. So we've had over 58 peer-reviewed publications since 2016, over 500 citations, and we've been either invited to or submitted and had accepted to multiple conferences, both nationally and international, over 100 presentations. So that's posters and oral presentations, as well as the publications, which I think are important in terms of creating this culture. The other footprint that we have is that in every province, we have an investment of dollars, people, and projects. And this is just a snapshot, if you will, across the country of the kind of dollar and personnel investment and the number of projects that are being either curated or participated in each of those provinces, which I think also helps very much with an understanding of what this network can accomplish. So the network has served as a hub to unite resources and develop new tools and infrastructure. It's really bolstering our capacity as a group of interested investigators to really do groundbreaking patient-centered research and ultimately to transform care and research in the real spirit of SPORE. We have strong partnerships with more than 30 organizations that include the provincial kidney care agencies, universities, and industry, and together have worked out a unique way of funding integrated research and clinical care and the translation and implementation in a way that I think should resonate with many of us and lessons can be learned. And so in summary, this conference is about innovation, and innovation is involving an improvement in the methods of working or producing whatever it is. And the CanSolve CKD network innovates through listening, learning, and leading to, from, and with our patients, caregivers, policymakers, researchers, and clinicians. And in that way, we believe is an example of a network as a tool to truly innovate. So thank you very much for your attention, and happy to answer questions at the end of this symposium.
Hello, my name is Kendall Ho. It's my distinct pleasure to be able to present at this Clinical Trials BC Modernization of Clinical Trial Conference. I'd like to speak with you in the next 10 minutes about telehealth innovation. And with this telehealth innovation, what do we need to do to modernize our trials so that we can actually demonstrate effectiveness and transformation of our health services? I love TV. And this series, The Office, is one of my favorites. I was listening to a podcast. They were discussing why is Office so special. They talked about several top reasons, but these are the three top ones. Number one, we get to know the characters deeply over the 9 seasons. Secondly, we're able to find beauty in everyday living, the ordinary, that we can see the blossoming of the flowers. And finally, each of the story arc, and at the end of the series, that we actually demonstrate growth of all the people in the end. These are what satisfy audiences. In the same way, when we think about clinical trials, these are also, I submit to you, important characteristics for us to demonstrate. Let me illustrate through telehealth. What I'd like to do is first talk about some of the principles about telehealth innovation trials. What do we look for? And then to speak to some of the current real-time virtual support in BC that I'm involved in as trials and also as evaluation. And then I love to go to the live session with you so we can have some Q&A together on this. Innovation Canada and many organizations use this technology readiness level, or TRL, as a way to evaluate innovation. Whereas TRL1 is you have vaporware. You have a concept, you think it's going to work. TRL3 is where you get to a proof of concept that you can start to demonstrate. People can start to see what you mean. By the time you get to TRL7, that you have a pilot project, you have a regional trial that you can actually start demonstrating what you do. And TRL 8 and 9 are really randomized controlled trial, multi-sites, so that you can actually demonstrate evidence. We often talk about in this journey from TRL 1 to 9, it's not even. It's not like every month you go through one. There are two specific what we call valleys of deaths. And the first one is from development to commercialization. This is where clinical trial really fits. How do you make sure that you have sufficient evaluation in the settings of the intended use? How do you make sure that end users are engaged? Health professionals, patients involved in that? And also, how do you make sure that there's alignments with what you do, with the health system gaps are? And then the second gap is about commercialization to rollouts. Now, that's a different talk for a different day. Focusing on clinical trials and now thinking about telehealth. This is a 2017 paper in New England Journal of Medicine, talking about the three kinds of telehealth. The first is about clinician to clinician, working together with technology like what we use to deliver care. Second is clinician to patients, so that we can directly see patients and patients sees us, that we can do interactions and do treatments. And the third is patients using technology like sensors or mobile phones to measure themselves, and then the resultant data is become useful for delivery of care. In that context, we have used the first two in BC to set up virtual care to support the COVID surge. In March, we have set up a group of us working with Ministry of Health. Together, you can see the organizations on the right side, setting up a total of 8 lines of real-time virtual support care in British Columbia. And these are disruptors in 2 clusters. The top half are health professionals and patients. For example, First Nations virtual doctors of the day, or later on, I'm going to talk a little bit about 811. The bottom half are the peer-to-peer support lines, which again, I'm going to highlight a little bit later on. Let's talk a little bit about one example of 811. This service is called HealthLink BC Emergency Eye Doctors and Assistants, or HIDE. Why did we set that up? Well, around March, because of COVID, a new disease, that there's a suddenly increased surge of volume of calls to 811. Also, each of the call became more complex because there's new knowledge. And so patients are very interested in knowing about the disease. Do they need to have it screened? Do they need to go to hospital? And in what circumstance do they want to consider that they are getting worse? There are a lot of emotional anxiety of our callers also. And so a group of us doctors were very kindly invited to work with our nurses, so our interprofessional team and 811 can manage the volume of calls to maintain strong access, to be able to discuss with our patients and direct them to either need to go to emergency or can stay home. So really appropriate use of our resources. And then also we want to be patient-centered. So when they need the information just in time, that we are there not only provide information, but to decrease the anxiety. And then finally, we want to make sure that this is integrated in a health service so patients can be referred back to their family doctors to be followed up. As we do this trial and do our evaluation, over this 8 months, we're able to find that with our service, that our daily volume with the 8-month period, we saw 16,962 patients. The wiggly line is the daily volume. That we found that it was averaging about 69 a day with a peak of 129. But then in fact, this past Christmas, December 26th was our highest volume per day, 139. And in January 4th, we've actually have gone to our 20,000 patients. We also look at the outcomes of these patients in triage. And we found that for those we triage, 3 out of 4 of them, we no longer need to refer them to emergency department within the 24 hours to see the doctors. In fact, we can reduce it, give them enough information so they can either stay home or follow up with a family doctor. We found that there's a further 16% that in fact we need to ask them to go to hospital faster. They have misperceived their indigestion and thinking that it's a GI tract problem, but in fact it's a heart attack. And so we need to accelerate them to go to emergency faster. And we found that patient satisfaction is very high, with 98% giving us 4 out of 5 or 5 out of 5 in rating. Now we look at a peer-to-peer side. Again, with the Heidi, we're using technology like phone, like Zoom, so that we can contact our patients, that we can use it very rapidly. In the same way, for this peer-to-peer, we use iPads that you can see on the picture on the left. And we also use Zoom and also regular technology that's not specialized. And we contact our colleagues anywhere in the province by mobile phones or iPads or desktop computers, so we can support patient care. And we have lines including emergency, ICU, pediatrics, maternity, and also mental health in this instance. And we found that over a 6-month period, when we evaluate, that we have calls coming from different parts of the province over time. And when we look at our volume, for example, in the emergency department one, that we can find the volume of call increasing rapidly over the 6-month period. It's continued to increase over time. And when you think about it, the 3 metaphors of what we try to generate with our real-time support is this, that we are safety nets so that no patients in the province will be left behind to receive care. Secondly, we want it to be a funnel so that wherever patients or health professionals work together, that we always go back to the longitudinal primary care system for the ultimate follow-up of the patient and longitudinal care that is patient-centered. And finally, we're looking at this as a fire station. Firemen need to put out fire, but in between, we build on relationship and build on communities throughout the province so we get to know each other so that this service become a friend to our patients, a friend to our colleagues. And so in the last 2 minutes, I'm going to talk a little bit about the evaluation of what we do. I think this photo really illustrates that. This is a group that wants to get fit, and so they want to learn together, dancing in front of the mirror, doing exercises with the coach. In the same way, when we do telehealth and we want to transform the health system, there's a system piece that we need to change. Peter Senge suggests that there are 3 very key principles of changing system. Number 1 is you need all the organizations to see the large picture together. And secondly, all the organizations having conversations and reflections, and then work towards co-creating the future. So this is what the system needs to do. But the individual organization and health professionals, we also need to self-evaluate to say, how do we contribute to the bigger picture? And how do we modify the way that deliver care? And then we need to keep monitoring ourselves as we monitor with others so that we can make sure that we can improve based on Bandura's, a Canadian psychologist, change theory. And so putting all together, when you look at that mirror and people dancing, it's really about 3 metaphors of evaluation. It's the GPS that help us set and get our goals agreed upon in the system. And so with that GPS set and the milestones, we then look at a mirror as data points, evaluation, to help it reflect on the actionable insights that we can learn from. But very importantly, we need to do the exercise, the PDSA cycle, the practice cycle that we need to introduce so that we can continue to do what we do and lead to understanding, are we getting closer to the GPS, to the destination where we want to go. And so coming full circle, this is why Office reflects very well on what we need to learn in evaluation, that we need to know our organization so that we know them deeply. We also need to use research trials to really unearth the beauty that the naked eye may not see, but in the background we see that these are the beauty of what the system changes is about. At the end, both on the summative and formative evaluation, that we see growth over time. And when we do see that, together with the people and the beauty, that's when transformation becomes a beautiful thing for us to see and experience in our health system. Thank you very much for your attention. I look forward to our live discussion so that we can have some dialogue together.
Many thanks for asking me to speak at this conference. I've been charged with the topic of innovations in pulmonary clinical trials. My name is Don Sin. I'm a respirologist at St. Paul's Hospital and also the director for the UBC Center for Heart-Lung Innovation, also at St. Paul's Hospital. There has been a tremendous impediment towards therapeutic development in respiratory diseases over the past 30 years. If we look at all the compounds that made it to phase 1 clinical trial and see what proportion made it to the clinic, in pulmonary medicine, less than 5%. In fact, only 3% of those compounds have made it to the clinic. This is in comparison to other therapeutic areas where larger successes have been noted. HIV/AIDS, the success rate is 14%. In neurology, it's 8%. Oncology, 7%. And cardiovascular therapeutics, the success rate is double that of pulmonary medicine at 6%. Most of these failures occur in early phase 2 trials. In fact, majority fail after 5 to 6 years of development in early phase 2 trials. And pulmonary therapeutics has been no exception to this rule. Over the past decade, big pharma have approached therapeutic development in a slightly different manner. One is to focus on genetics. And what they've shown is that drugs that have linkages to human genetics have about 50 to 100% increased success rate of passing through early phase 2. Overall success rate is in the range of 70 to 80%. However, if the therapeutic target has no genetic linkage based on genome-wide association studies, the success rate drops to around 40%. Even a better method of improving the success rate in early phase 2 trial is to find response biomarkers or efficacy biomarkers linked with the therapeutic product. In such cases, the success rate jumps up to well over 80%. In contrast, for therapeutics that do not have a response or efficacy biomarker, the success rate is below 30%. In pulmonary medicine, the recent randomized controlled trials, especially for early phase 2, have focused on utilizing these novel biomarkers. One advantage that pulmonary medicine has is that there is a robust intermediate readout. It's called forced expiratory volume in 1 second, or FEV1. And this value can be obtained from simple spirometry, as shown here. Spirometry is inexpensive, it's rapidly conducted, takes only about 15 to 20 minutes, and produces reliable and robust data in terms of lung function. And lung function parameter is measured with this output, where volume is plotted on the x-axis and time is plotted on the x-axis, and volume is plotted on the y-axis. And this is the expiratory part of the flow volume loop. And you can see in healthy individuals, it can expel 5 to 6 liters of air. In asthmatics or those with other related obstructive airways disease, such as COPD or cystic fibrosis, there is a diminution amount of the volume of air that can be expelled, especially over 1 second. And that's denoted here in the black line. However, FEV1 alone is relatively insensitive to detect therapeutic changes for drug development. And as such, FEV1 has now been modified slightly to make it work for early phase 2 trials. And such modification is shown here in this early phase 2 trial conducted by Gail Gavreau and others at McMaster University. The investigators wanted to know whether AMG 157, an antibody against a novel therapeutic target that was discovered through genetic linkages studies called TSLP, whether antagonization of TSLP or inhibition of TSLP can improve asthma symptoms and other parameters. To conduct this study, what the investigators did was expose participants to allergens. So asthmatics are generally atopic and allergic to a variety of different aeroallergens. And so after an allergen challenge, the investigators then asked the participants to perform spirometry and generate FEV1. And they did this on multiple occasions throughout the 12-week treatment period. And this type of data were generated to show the therapeutic promise of this compound. With the allergen challenge, there is a rapid diminution of FEV1 by about 30% decrease relative to baseline among those receiving placebo. However, among asthmatics receiving the therapeutic product AMG 157 anti-TSLP antibody, the diminution was cut by almost 50%, only about 17% in absolute terms. These data suggested that anti-TSLP could work in preventing lung function reduction, and most importantly, reducing patient symptoms and exacerbation among severe asthmatics. This product is now in phase 3 trials, and the readout is expected in 2021. For phase 3 trials, pulmonary investigators have now adopted an adaptive design for large randomized controlled trials. Here we define adaptive design as one that allows for plan modifications to one or more aspects of the RCT methodology based on accumulating data from subjects in the trial. An example of an adaptive RCT for a bronchodilator is shown here. The bronchodilator in question is indacaterol, which is an ultra-long anti-spasmodic and induces bronchodilation. Tiotropium is the standard bronchodilator that is used for COPD patients. So the investigators asked this very simple question: does indacaterol improve lung function? And what is the optimal dose of indacaterol that can be used? To answer these two important questions, the investigators used an adaptive trial design. The first phase was testing the 4 different therapeutic doses of indacaterol, ranging from 75 micrograms per day to 600 micrograms per day, and in comparison to standard bronchodilators, formoterol and tiotropium, as well as a placebo. And this was done for 2 weeks, and their enrollment target was 770 patients. The readout was FEV1. And what they showed was that there was dose optimization between 150 and 300 in terms of FEV1 improvement. After this first phase, they then carried on with the optimal doses for a longer 26-week therapeutic trial, where they compared 150 and 300 microgram doses to tiotropium, which is the standard bronchodilator, as well as placebo, and looked at lung function over this period. And what they showed ultimately in both those aged less than 65 and those above 65 was that indacaterol in 150 to 300 doses was superior to tiotropium in terms of inducing bronchodilation. Adaptive design trial designs are being used increasingly not only in COPD but in other pulmonary disease areas such as cystic fibrosis, pulmonary arterial hypertension, tuberculosis, and it's more recently it has been used to assess therapeutics in COVID-19 trials. And we saw one recently from the Solidarity Group run by the World Health Organization. So in summary, pulmonary clinical trials are now increasingly using genetics to identify novel therapeutic targets. They're also using novel biomarkers to assess very promising trials, especially for phase 2A therapeutics. Spirometry, FEV1 alone is too insensitive for that purpose. So there has been modification to FEV1 on how it is used. And for phase 3 trials, adaptive trial designs are increasingly used to address multiple objectives and to ensure that the study has sufficient power to answer multiple endpoints and hypotheses. And with that, I'll be happy to entertain any questions that you may have. Thank you very much.
So I'm Rob McMaster. Really like to thank our 6 speakers for outstanding presentations. And what we didn't have a discussion beforehand of commonality across the different areas. And as you can see, many of the innovations we see in different areas. And I guess some of the questions that are coming up is how applicable are, say, adaptive trials to other areas of research. And I think we can see from Howard's talk and from Dawn that is already transpiring across other areas. Similarly, I think with Adira, there's some questions coming up on networks. I believe the value of forming these chronic disease networks and how is it a model for other chronic diseases? We just heard about asthma or COPD were some of the questions. I think telehealth clearly probably has applicability in many different areas of clinical trials. And then we have difference in numbers of whether it's using imaging in MS or N-of-1 trials for rare diseases. I mean, there's a number of different innovations required across the spectrum. So we now have a few minutes for discussion. And there are a few questions from the audience already. I can read some of them out to different, and I think we could take a stab at some of these questions. One of them from Bernal Eigel is regarding adaptive trials is what's the response from regulatories on when you propose a trial? And I guess either Dawn or Howard could take that one. Maybe Howard could discuss it first. It's, how is it difficult to get approval, either at the ethics or at others or FDA Health Canada for adaptive trials? And is there an additional cost for it? Those are the 2 types of questions coming forward.
You're muted.
Naomi, we have no sound here.
Me?
Yeah, thank you. Oh, good.
Thank you. So regulators prefer to see pre-specified rules of how the trial will run, and adaptive trials by their definition are going to adapt in the course of the trial. So there's a gap that has to be filled in order for this to be successful. Now, I think it's possible insofar as presenting the modeling and the algorithms that are going to be used. And the critical thing with adaptive trials is that they be pre-specified so that you don't make up the rules as you go along, but you apply rules that you've preset and then they get applied during the trial. So it's possible to navigate regulatory, but it's not a step that should be missed. It has to be pre-planned and is quite important. In relationship to the additional costs, there's a lot of back-end modeling and simulation because a lot of the algorithms that guide the adaptive randomization and other things are pre-specified based on modeling and simulation of cohorts with the disorder that serve as reference for the trial. And that's how the rules get set. So that is much deeper than it is typically in a traditional design.
And a similar question to Sylvie is sort of, for N of 1 studies, again, what is the regulatory aspects of that? Are they difficult to get approved? Do they take a different type of approach?
Well, I think currently N of 1 trials are not well known yet, and many people still think it's just simple case reports. So we have to do a lot of educational work to make people understand what are N of 1 trials. Currently, they are used mainly for proof of principle studies for innovative therapies, which can inform larger group trials. In terms of regulatory authorities, I know that these N of 1 trials, particularly those who provide meta-analysis, are considered, but I'm not aware of a true example.
Thank you. I guess from the six presentations, do you have questions to each other on different areas? And as we were trying to see, there's such an overlap as we would want on trial design and applicability to different diseases, different symptoms, or different areas.
Rob, I have a question that might be fun for Dawn to answer. So one of the things about adaptive trials is discrete outcome measures. So in the example that I chose of migraine's a good example because it's, you're binary. Either your headache responds or doesn't respond. I wonder what the experience in pulmonary medicine has been around setting those outcomes for adaptive trials and how discrete you've been able to make them, and what your level of contentedness is with the decision points.
So thank you for the question. So certainly in COPD trials, there are two main metrics that regulatory agencies and others consider. One is the lung function measurement that I mentioned, FEV1, and the other is exacerbation. And exacerbation is a binomial distributed type of parameter, whereas FEV1 is a continuous measure. FDA, for instance, takes FEV1 not as a continuous measure, but as a dichotomous. So you have to, a therapeutic product has to achieve a certain threshold in order to be clinically relevant. And that really depends on the disease state. In COPD, it's about 100 mLs. So that's one approach that regulatory agencies take with these adaptive designs for sponsors to demonstrate such an improvement in their underlying exacerbation in FEV1. As for exacerbation, they would insist on a binomial distribution in terms of the analysis that's done at the end. And again, I think it was well pointed out that this has to be pre-specified and work with the FDA to ensure that the analytic plan is well within what they would consider acceptable even in an adaptive trial design.
Thanks, Don. So we just have a few minutes left, and I just wanted to highlight one of the slides that Kendall showed of valleys of death. And clearly, clinical trials going to address, as Kendall pointed out, in some instances, that first valley of really getting the information to move forward. And then the second valley that we are all so well aware of in translation is how do we implement it into practice, across at scale, I think we're calling it now. So how about if we have 30 seconds each from each speaker to talk about not the barriers, but how, what are the successes? What do we take to have success, to have from clinical trials into clinical care or into common practice, whether it's prevention or treatment? I'll just start in the order that we were. On my screen, I see Howard. So I'll put you on the spot for this. How do we move it forward into practice?
So Rob, I think one of the most important factors that we have to address is when to recognize that a treatment is not going to work and how to reduce opportunity costs. So in the work that we do, it's trying to be as efficient as possible, and that involves killing programs and being decisive and not wasting resources on programs that aren't going to work out, and yet not losing the opportunities, not declaring them dead prematurely. And I see the largest challenges and the most amount of money wasted on programs like that.
Good, thank you. We actually have about 10 minutes left, so you can expand your answers to more than a minute. Tony, I know each of you have addressed, throughout your talk, but I mean, it is that really pushing it into practice, as opposed to clinical research.
Yeah, and I think as I listened to all the presentations so far in the concept of innovation, one area that's really interested me is how do we repurpose existing medications with our new innovations, can we efficiently design a study or identify medications that could be beneficial for a disease that weren't previously thought of? And I was thinking of Don's presentation using genetics or GWAS. Could that help us screen a panel of existing medications to better identify and have that high success rate of the 70% plus success rate? And then using Howard's concepts of innovative trial design to do it as local investigators, because it's very expensive even to do your own local study. That's another challenge. And then how can we integrate that maybe with Adira's infrastructure, with the SPORE infrastructure? Can that be leveraged into success as a community? So that would be my fantasy of where we go forward with a variety of diseases.
Thanks, Tony. Sylvie?
Well, I think in rare diseases, we always have been acting in the interface between research, clinical trial, and real-world clinical practice. And I personally really think that the end of one trial philosophy will be something that will help us to evaluate the efficacy of treatments in clinical practice in the future. So rather, as a doctor, sitting in the office and asking the patient if it helped or it did not help, really to work with a pre-made protocol to ask the patient and to evaluate whether the treatment works for the individual patient or not. So that would be my take-home message.
Thank you. Adira, you did address it. Basically your whole talk was about implementation, but.
Well, yeah, I think what we've learned is the more patients that you engage both in the trial and then what we're also working on is an implementation sort of strategy, if you will, so that the investigator is also learning how to do briefing notes and how to actually engage policymakers and organizations for when the study is finished, and the value proposition of why this either does work or doesn't work. And to Howard's point, if you discover that things don't work, we need to stop doing things in medicine that are costly. And so, part of, I think, what's really interesting to us is that patients actually sat in a room and said to us, do you mean that there are medicines that actually work and you don't have to give them to us as a doctor? They were shocked. They had no idea that it was physicians' choices at the end as to whether or not they prescribe drugs that work. And that shocked them. And so I think the more you can change culture, like HIV, and Don or Howard, I can't remember who did the slide, but the HIV, the fact that there's more drugs in play, successful, may very well be because they've always engaged the population at the very beginning, from both study design testing, full recruitment, 100% engagement, and then implementation. So maybe those are lessons learned for us as we think about these diseases and things that are more in common than different about how to change that landscape. Those are my thoughts.
Kendall?
Yeah.
Your slide I was referring to.
Yeah, I appreciate that. In fact, I was thinking of kind of four steps I can think of how we can use trials to leverage and improve and implement. One is to really kind of focus on a clinical gap. And in that gap, how can either medications or technologies can solve that gap? And I think that will be very important both at the clinicians, the technology or the drug companies, and the patients together to actually look at that gap. And health system needs to be there all the time. But in this particular case, I think maybe in the background a bit. But then secondly, the second step is, well, how do we collaborate and co-create a potential solution? Again, it needs clinicians, patients, and the companies. The third is a very important one, which is really vital for trials, is to set up the right methods to generate the evidence, be it N of 1, advanced trials, or usability, etc. The evidence is going to be very important. I think the final step is one of transformation. That's change management. I think just because we have evidence does not lead to transformation. And so how do we use networks like what Adira talked about so that we can actually engage policymakers, patients, clinicians, and all the actors together to do that transformation? So I think those four steps would be important for us to get through the curve to bring an innovation into practice.
Thanks, Kendall. Don, I know this is an area that you're actively involved in.
Yeah, with variable success.
Fair enough.
I was really, impressed by Adira's ability to create a network that actually has stakeholder engagement, including the patients. And I think this is critically missing, certainly in many pulmonary therapeutic areas, where we don't engage the end user. We don't even engage the community of physicians when we design therapeutic trials. So it's not surprising that even with positive trial results, very little of that is, adopted into the community. So I think the lesson I've learned from today is that we really need to build a different type of network in designing therapeutic trials, one that includes stakeholder groups, including the patients. The other, I was very impressed with Sylvia's talk about N of 1 trial. And in fact, pulmonary medicine was one of the early adopters of N of 1 trial. Gord Guyot did a therapeutic N of 1 trial published in New England Journal in the 80s, debunking the use of theophylline for COPD. And for some reason that sort of went out of favor in pulmonary medicine. But I think we need to bring it back, particularly for a lot of therapeutics that don't have strong RCT data. A lot of therapeutic products that don't have strong RCT data behind it, some of the legacy therapeutics that we're using. So I was very impressed with that as well, and we should be doing more of that as we go forward.
Good. I think we've all had I've certainly learned a lot from the morning's session. There's a lot of commonalities, challenges, and I think we've seen some very different approaches to finding solutions. There's a few more questions coming from the audience. One is on adaptive trials, which is, what is the patient's perspective on it? I guess of knowing that they may be, I'm just interpreting, they may be on a dose that is not optimal, but it's part of a clinical trial. Howard, you've had a lot of experience.
Well, I think the explanation within an adaptive trial should be more participant-centric, because the whole idea is that through a technique like adaptive randomization, you're lessening the likelihood that inefficient doses will be tested throughout the length of a trial. And that's by contrast to a parallel fixed design where you're assigned to that group, nothing's going to change it. So your odds of benefiting by playing the winners and dropping the losers actually is better in an adaptive trial. So I think from a participant viewpoint, that should be an attractive alternative.
Good, thank you. And I think maybe the last question is Heidi Scott, it's been a topic that I know is coming up throughout the next, well, 2.5 days now. Is how do we get the results of clinical trials back to the patients and back to the public that participated in them? And I know we've talked about this a lot on Clinical Trials BC, and Adira had some solutions. But I do think, that is an area that we probably need to improve on, really to keep that engagement long term. Any thoughts on Adira? You've spent the whole talk.
I mean, I think we need to practice speaking in lay language. I think people understand things if you tell them simply, and that is no matter how educated they are. So I think that certainly having patients who are in the study help to be part of that messaging is another really good way to get to keep the message going, and then they'll be excited to be in the next clinical trial, so it builds a group. And then having various means to distribute. I think we need to tell people who were in studies and those who were not what the results were. That they understand. And I think we need to commit to that and not just give it lip service. We have patients and partners reading lay summaries and giving feedback to everybody about whether it's understandable or not, that kind of thing. And so I think that we all need to learn how to do it better. So I think education around better communication is important.
And
Rob, can I jump in on that one? I absolutely agree with what Adira said.
With telemedicine, is it easier or more difficult?
In fact, I think it's very important to give feedback not only on the evidence itself, but really is at the end of the day what I think Adira very successfully do is build the relationships with the patients. At the end of the day, it's that relationship, be it the researchers working with each other, health policymakers, and in this particular case, our citizens and patients. And so the fact that we share evidence, the fact that we support, understand the patient's perspective of gaps, every one of them is a relationship building exercise. And in this relationship building, the trust is where authentic feedback comes back. So I really support the fact that we do need to give the evidence back. And also the patients themselves would have networks to distribute them. But if we go forward, I think that relationship will be a very important piece to build.
Thank you, Ken. And I think that's a very good place to end our discussion on it, is that feedback. So what can I say? It's been a very innovative morning. And the next session is on cancer innovations in cancer clinical trials, which I think starts in 15 minutes. And we hope to see additional innovations and see what's in common from what we talked about this morning. So again, thank you very much for all the speakers this morning. And I'm glad we have our technology working, and really look forward to the other sessions throughout today, tomorrow, and Thursday. Thank you very much again. Thanks.
Thank you.
Thank you. Bye-bye. Enjoyed it a lot.
I thought that went very well.